my cutest little champions, I welcome you all to solve top 30 questions from the chapter Sexual Reproduction in Flowering Plants, and this is for KCET 2025. Other children also who are preparing for some other exams, okay, you can also give a try. It's really good for you to have an exposure to MCQ type of questions. Yes, so let's begin. But the rule, I hope everyone remembers. What's the rule? You have to pause the video, try to find the answer by yourself, and then only listen to my explanation, or else we are not actually checking our preparation. Right? Yeah. So the first question that we are having over here is fresh. A flower has 10 stamens. How many stamens? 10 stamens. Okay, cool. Now, each having bilobed dithecous anther. Bilobed dithecous as in you have two lobes in an anther. This is how it looks. Okay. This is one lobe. This is second lobe. This is dithecous as in this is one theca. This is second theca. Over here, this is also dithecous. One theca, second theca. Okay. Now, if each microsporangium has 5 pollen mother cell. Okay, each microsporangium has 5 pollen mother cell. Listen to me. How many microsporangium are there in an anther, one particular anther? So if you see, this is an anther. Over here, you will be having one microsporangium, second microsporangium, third microsporangium, fourth microsporangium. So how many microsporangium in total? In one, 4 microsporangium. Okay, now, if each microsporangium has 5 pollen mother cells, there are how many mother cells? 5 pollen mother cells. Over here you have 5. Okay. Now, how many pollen grains would be produced by the flower? Okay. One pollen mother cell produces 4 pollens at the end. Okay, 4 pollens or 4 microspores actually. But yes, you can write 4 pollens at the end. Cool. If one is producing 4, then how many will be the 5 of them will producing? How many will 5 of them produce, children? In one, if you have 5 of them and 5 of them are going to produce 4 pollens each, you will be having 20 over here. How many, children? There will be 20 pollen grains produced by this. There will be 20 produced by this, 20 produced by this, 20 produced by this. So can I say 20 into 4? Will 20 into 4 work for one anther? For one anther, can I say there are 4 microsporangium, right? And if each microsporangium is giving you 20 pollens, so 20 into 4 becomes what? 80. 80 pollens are there in total. Now, you have 10 stamens like this. 10 stamen means 10 of them will be having anther, right? So 80 into 10 will be becoming what? 800. It becomes what? 800. This is the answer. Please remember, in each anther, you will be having 4 microsporangium. That might not be given in your question, but you know it. Now, each microsporangium, if it has 5 pollen mother cells, okay, if it has 5 pollen mother cells, if one pollen mother cell gives to 4 pollen grains, then 5 pollen mother cells, 5 into 4 gives you 20 pollen grains in one. So 20 into 4 becomes what? 80. And how many stamens do you have? You have 10. So it becomes 800. This is the answer. This came in 2023. Works? Interesting, no? Yes. Now, primary endosperm nucleus is formed by the fusion of what? What do you mean by primary endosperm nucleus? So before I go ahead with this particular topic, I want to tell you something very much important. That is about the female reproductive system of a plant. Listen to me very carefully. Okay? And then you will be able to solve so many questions and answers. I won't be explaining again, okay? So over here, sorry, forgive me. Okay, let me make it over here. So over here, this is the stigma, this is the style, and this is the ovary. Cool. Now, in the ovary, you have something that is known as ovule. Okay. Now, I am going to draw it separately. So, this is your ovary, children. Inside the ovary, you have the ovule. This is the ovule. Okay, or probably I can make a bigger, oh, it's okay, fine. Over here, the ovary has something which is known as placenta, and the placenta is attached to the ovule with the help of something that is known as funicle, and this point of attachment is known as hilum. This provides the initial nourishment. Now, the ovule is surrounded by two layers, protective layers. One layer goes like this, and then you have another layer which will be going like this. They are what? Integuments, which is there for protection. Okay, this end, it's open. This open end, I will call it micropylar end. Okay, the opposite to micropylar end, whatever region comes, it is known as chalazal end. Opposite, wherever you find micropyle, opposite to it you will find chalazal end. It's not like this particular funicle placenta will be always there in the chalazal end. No, this can shift to this side also. The funicle and placenta can be here. Still, the side opposite to the micropyle will be known as chalazal end. Now, what is in there in the ovule? In the ovule, you have so many cells which are having reserved food materials. It is filled with cells. And what do you call this cell as? Total, all of them together, you call them as nucellus. You call them as nucellus. Okay. Now, one among this nucellus, it is covered. Okay, I am not making the whole thing. One among this nucellus becomes what? The megaspore mother cell. What does it become? Megaspore mother cell. This ovule that you see over here, from here to here, this is known as megasporangium. This is known as megasporangium because this is where everything is formed. The megaspore is being formed. Okay. Now, this is megaspore mother cell. One cell among them. This is the megaspore mother cell. It will undergo meiosis. How many number of chromosomes does it have? Double. 2n number of chromosomes, normal number of chromosomes that every cell has. It will undergo meiosis. This is meiosis 1. Meiosis occurs in 2 cycles. At the end of meiosis 1, you get 2 cells with half the number of chromosomes. This will be haploid. Again, meiosis 2 has to happen. This will divide again. This time will it reduce? No, children. It will be all haploid. Okay. This will be still haploid. So, the nucleus that you see, it is diploid or haploid? It is diploid. It is diploid. One cell among them is megaspore mother cell, right? So, all of them, they are basically diploid. Please remember this. 2n means diploid. Now, 3 of them, they degenerate. 3 of them, they degenerate. One guy stays alive. And this becomes the functional megaspore. This becomes the functional megaspore. The story has not ended as of now. I have to go for the next thing only. Okay. So, functional megaspore is there. Now, I am going to tell about functional megaspore. So, I have formed a functional megaspore over here. Let's see what happens with functional megaspore. Suppose this is the functional megaspore. The functional megaspore has its own nucleus, which is haploid nucleus. This haploid nucleus undergoes one division, making it, there will be 2 nucleus. One division happens, this nucleus divides into 2. Okay. Again, second division happens, this nucleus divides into 2. Once more division will happen, this nucleus divides, and at the end, how many nucleus will you get? 1, 2, 3, 4, 5, 6, 7, 8. You get 8 nuclei at the end. 3 nuclear division happens over here. It is not meiotic division, okay? Just the nucleus is dividing mitotically. So, 3 nuclear division happens. At the end, you get 8 nucleus. At the end, how many nucleus you get, children? 8 nucleus. And I am going to place the 8 nucleus like this. Your 8 nucleus, no. Suppose this is 1 nucleus, this is another, this is another. This is 1, this is another, this is another, this is another, this is another. I place the 8 nucleus. Now, the 8 nucleus are surrounded by cell wall, cell membrane. So, this becomes 1 cell, this becomes another, this becomes another. Okay, 3 cells you got at the top. This becomes 1 cell, this becomes 1 cell, this becomes 1 cell. This becomes another cells. These 2 nuclei stays like that only. And I will now name them. This cell which is formed over here, no, this particular thing, how will I show? I will make it in red. Okay. This cell is known as the egg cell. Okay. Opposite to the egg cell, you have two cells, one over here, one over here. You call them synergids. These are the best friends of the egg cell. These are known as synergids. Okay. These three cells which are right over here, three of them, they are known as antipodal cells. Antipodal cells. Why I am explaining it to you is because so many questions will be coming along with it. So these are known as antipodal cells. These two nuclei which is present over here, it is known as polar nuclei, two polar nuclei. And this is a part of a cell. This area that you see is a cell which you call as, so polar nuclei is a part of central cell. Okay. So how many cells are there? 1, 2, 3, 4, 5, 6. 6 cells are there, and then this is one big cell. So 7 cells are there. How many nuclei are there? 1, 2, 3, 4, 5, 6, 7, 8. 8 nuclei are there and 7 cells are there. So this is a condition. 8 nuclei are there. Number of nucleus is more. The cell is less. There is one cell less. This thing that you see is central cell. It has 2 nuclei. So you have 8 nuclei but 7 cells. Okay, please remember that. Now, the thing is, when the pollen tube comes along with the pollen tube comes, suppose it has 2 male gametes. The pollen tube always carries 2 male gametes. One male gamete goes and fuses with the egg. One male gamete goes and fuses with the egg cell. Male gamete is haploid, egg is also haploid. You get diploid zygote. What do you get? You get diploid zygote, children. Okay, you got the zygote. Now the next, the male gamete which is there, it goes and fuses with the 2 polar nuclei which is present over here. So next male gamete, which is again N, fuses with what? 2 polar nuclei. Now polar nuclei, if we say, one is N, other one will also be N. So N, this has now become triploid cell. So now the thing that you get is known as triploid endosperm nucleus. This is triploid primary endosperm nucleus. What do you call this as? Whatever is formed is known as primary endosperm nucleus, and it will be triploid, my dear children. So all the cells which were present over here, they were all haploid. One guy came in, did diploid zygote. Other guy came in, fused with the 2 polar nuclei, made it triploid. Not even diploid, triploid primary endosperm nucleus. This primary endosperm nucleus, why do you think this is formed? To provide nourishment to the zygote. It provides nourishment to the zygote. So that zygote develops into what? Embryo. Okay, cool. Now, from here, we are going to start solving so many questions related to megasporangium and all this megasporogenesis and stuff like that. So I hope you got this. If you have not got this, pause the video, try to understand this picture and then only go ahead. Okay. This process where fertilization is happening twice, it is known as double fertilization. What is it known as? It is known as double fertilization. And over here, 1, 2, 3 nucleus are coming together. It is known as triple fusion. This thing is known as triple fusion. This thing is known as syngamy. Works? Okay? Chalo. So primary endosperm nucleus is formed by the fusion of. How does primary endosperm nucleus form, children? You saw what it is fusion? 2 polar nuclei and 1 male gamete. So 2 polar nuclei and 1 male gamete. This becomes the answer. Cool? Next question. Which pair of the following cells in the embryo sac are destined to change their ploidy after fertilization? So who will change its ploidy after fertilization? In the embryo sac, all of them are haploid. There are the 2 cells which change its ploidy after fertilization. One is egg cell because it will undergo fertilization with the male gamete to form diploid zygote. One is central cell which has 2 polar nuclei which will be fusing with another male gamete forming triploid primary endosperm nucleus. So this becomes your answer, 2023. This is why I told understand the concepts better. Okay. Next, stalk of the stamen is known as. In the stamen, this is the structure of the stamen. Over here, this stalk-like structure, what do you call it? You call this as filament. Okay, this is the anther. Cool. So this becomes your answer. Next question. The ovule of angiosperm is technically known as. The ovule is known as megasporangium, because this is where the megaspores are going to develop. What do you call it? Megasporangium. Okay, next, typical mature embryo sac of angiosperm is, how many nucleus is that? How many cell it has? Cell, the number of cell is less, nucleus is more. Okay, so it will be 8 nucleated, but it will be 7 celled. 8 nucleated, but it will be 7 celled. Okay, because 2 nuclei are being taken by the central cell itself. You now see how many questions you are able to solve with that particular topic. Yeah. Next, one of the 2000-year-old viable seed discovered during the archaeological excavation at King's Herods. It's not Herod, it's Herods, near Dead Sea was. So there was 2000-year-old viable seed which was still probably able to germinate and stuff like that. So what was that particular seed belonging to? It was belonging to date palm, a date palm. But do you know the name? Any guesses with the name? It is known as Phoenix dactylifera. This is important. 2000, whenever it comes, just understand it is Phoenix dactylifera. If the question comes for 10,000, okay, if the question comes for 10,000 and Arctic region and stuff like that, it will be lupinus. That is something else. So 2000 always for Phoenix dactylifera. Next question. Identify the correct order of events in the pollen pistil interaction from the options given below. Okay, so pollen pistil interaction means this is stigma, this is a style, and this is the ovary. Inside the ovary you have ovule. Inside the ovule, what do you have, children? You have the embryo sac. Okay, so pollen, when it lands, okay, so first will be deposition of pollen grains on the stigma, release of male gametes into the embryo sac, entry of pollen tube, development of pollen tube, entry of pollen tube. Okay, deposition of pollen grains on the stigma, first thing. Now once it has deposited, it will form a tube. So next will be pollen tube formation. So entry of pollen tube into the embryo sac, development of pollen tube, this becomes the second. Okay, so we have first this is second. Now development of pollen tube, after the pollen tube has been formed, where it has to get inside, children? It has to get inside the ovule. It has to get inside the ovule. So entry of pollen tube into the ovule, this become third. Okay, then entry of pollen tube into the embryo sac. After it gets into the ovule, it will be getting into the embryo sac, right? So this will be the fourth one. And then release of male gametes into the embryo sac, this will be the fifth one. So let's see, 2, 4, 2, 4. So this and this becomes cancelled because 2 and 4 is not there. Now 2, 4, 5, 2, 4, 5, 3, 1. This becomes the answer. Okay, 2, 4, 5, 5, right? Yes, 5, 3, 1. This is the answer. Okay, please always remember, inside the ovule you will be having your embryo sac. So ovule will be coming first, then the embryo sac. Cool. Next, how many microsporangia are located at the corners of typical bilobed anther of angiosperms? Just to make you confused, how many microsporangia are there in a bilobed anther? In a bilobed anther, you saw 4 microsporangia are there, 1 in each corner. Okay, so basically it becomes 4. So your answer is 4. Don't get confused. 2021 question. Next question. In the given diagram, identify the parts labeled as A, B, C, D. Okay, so this given diagram is of monocot embryo. Monocot as in grass and stuff like that. Okay, castor, coconut, they're all monocots. They just have one cotyledon. So what happens? The primary endosperm nucleus you saw, no, it forms endosperm. The endosperm provides nourishment to the zygote. But the zygote, when it's in the growing stage and it forms an embryo, it will be like, I don't need endosperm now. I would have my own nourishment thingy. I will call it cotyledon. I will call it cotyledon. So basically the endosperm, some of it get transformed into cotyledon. So there are some type of plants where whole endosperm is taken up to make cotyledons, and they are known as dicots. They will make 2 cotyledons out of it. Okay, there are some plants who will say that let some endosperm persist, I will make 1 cotyledon. So that is monocot, which has 1 cotyledon. And how do they look? So basically it has an embryonal axis. To make things easy for you, I will make it. So it has an embryonal axis which goes like this, children. This is the axis of the embryo. Okay, from this point it will form something like this, This is one cotyledon that it has, and this cotyledon is known as scutellum. This is the only cotyledon it has. Now the scutellum is covered by the persisting level of endosperm. The endosperm is still there, little amount I told you, right? So endosperm is there. Now from this point, you saw the attachment, upper point, no? At the tip you have certain region which gives rise to the shoot, and this is known as plumule. Plumule is surrounded by certain tissue which is known as coleoptile. What do you call it? Coleoptile. Over here at the end you have the root tissue that is known as radicle. What do you call it? Radicle. Now the radicle is also covered by something that is known as root cap. Now basically there will be things to cover this also. Okay, and what do you call this as? You call this as coleorhiza. What do you call it? You call it coleorhiza. So this is the structure. This is the whole structure. Now over here A is mentioned, which has things like this, just like how I made. It will be what? A will be what? Coleoptile. D will be what? D will be coleorhiza. So look for that. Coleoptile, coleorhiza. Anywhere it is there? No. So coleoptile, coleorhiza is there. Now B is a region that comes from here, is a region that comes between these things, that is the scutellum. So B becomes your scutellum. C thing that you see over here, it is the outermost layer. So basically this is the embryo. This is the embryo which is forming inside the ovule. Okay. So your ovule, in the ovule all this thing is happening, right children? In the ovule all this thing is happening. Outside the ovule what do you have? You have, outside the ovule you have the ovary. And the outer layer of the ovary becomes the outer layer of the fruit. So if this particular guy, that is the ovule, if it becomes the seed, then this ovary becomes the fruit. So the outer layer of this will be known as pericarp. What do you call it? Call it pericarp. So it was ovary first. Inside the ovary you had the ovule. After fertilization, inside the ovule the seed is being formed. Basically the zygote is developing into embryo, and this ovary becomes the fruit. So the outer wall is known as pericarp. Okay, so this becomes your answer. So if you are not aware about these arrows and stuff like that, it will be difficult for you. That is why I helped you understand that. Okay, works. Next question. Consider the following statements and choose the correct answer from the given options. Statement 1. Innermost layer of microsporangium is known as tapetum. Microsporangium is known as tapetum. Cells of tapetum possesses dense cytoplasm, more than one nucleus, and nourishes the developing pollen grain. So the microsporangium, how does it look? So I told you. Okay. So this is the anther that we are having, okay, which I messed up here. Wait. Ayo. Okay. Okay. Fine. So there are microsporangium, 4 of them in the corner. The microsporangium, no, there is a layer over here which is known as the tapetum. So tapetum is there, and tapetum definitely provides nourishment to this particular thing, the pollen grains which are going to get formed over here. Outside the tapetum, what do you have? If this is the innermost layer, there might be outer layers also. So the outermost layer that is there, it is known as epidermis. Okay. Then you have one more layer which you call as endothecium. Okay. Then you have one more layer which comes between this. This will be known as middle layer. And the innermost layer is tapetum. Tapetum, as you can see over here, the cells of the tapetum possess dense cytoplasm. Yes, it does. and it has more than one nucleus? Definitely yes. So it provides, it's the only layer that provides nourishment, whereas these 3 layers provides protections. It provides protection and helps in dehiscence of anther, as in breakdown of anther. Works? So both the statements, as you can see over here, are completely true. So both 1 and 2 statements are correct. Cool? Okay. And this cells, no, there are group of cells over here, you call them sporogenous tissue. And each sporogenous tissue, each cell of the sporogenous tissue is known as microspore mother cell, because they can undergo division to form microspore. And they will be forming 4 cells. In megasporangium you saw 4 cells are being formed, but 3 of them degenerates. But over here 4 of them will be giving rise to 4 pollen grains. So identify the correct statement. Only one megaspore present towards chalazal end remains functional. So chalazal end is the downward end, micropellar is the upward end. Always in the micropellar end only you will be finding the egg cell. You will be finding the egg cell. Okay. So only one megaspore present towards the chalazal end remains functional. No children. This is not true. Okay. It is not remaining near the, there is only one functional megaspore, but it is not remaining near the chalazal end. It stays near the micropellar end. Okay. Okay. Now, no, it's not defined. It is not defined actually. I'm so sorry. Okay. Now, 3 megaspores present towards chalazal end degenerate gradually. Is it like that? No. Each megaspore mother cell directly develops into a megaspore. Does it directly develop? No. Wait. I have. Next question. Identify the correct statement. Only one megaspore present towards chalazal end remains functional. B. 3 megaspores present towards chalazal end degenerates gradually. Each megaspore mother cell directly develops into a megaspore. No. Each female gametophyte is 7-celled and 7 nucleated structure. No. It is 8-celled and 7 nucleated structure. Now, only one megaspore present towards chalazal end remains functional. No. 3 megaspores present towards chalazal end, they degenerate gradually. This is the correct statement. Okay, so these 3 will be degenerating, rest 1 will be surviving. Next, which one of the following aquatic plant does not show pollination by water? So pollination by water, as in the pollen grains will be traveling through water and the pollination would be happening. So Vallisneria is one such where things will be happening. Hydrilla is one such where water pollination is happening. Zostera is definitely one such where water pollination is happening. There is water hyacinth, which gets pollinated with the help of wind and stuff like that, but not water. Okay, so water hyacinth has water in it, but still it is not going to get pollinated with the help of water. Okay, so this is the one that does not show pollination by water. Next question. Which cell of the female gametophyte is involved in the formation of primary endosperm nucleus after fertilization? So you know it. Primary endosperm nucleus means you're talking about central cell. The 2 polar nuclei of the central cell plus the male gamete gives you primary endosperm nucleus. You tell me how easy have it become for you to now understand things since we drew that. Now next question. In apple, the chromosome number of the gamete is number of chromosome, okay? How many? 17. And remember, this is a gamete. And gamete always have haploid content. It always have haploid content. What is the chromosome number in the primary endosperm nucleus? If haploid is this, okay, and primary endosperm nucleus, then it has to be triploid. It has to be triploid. If haploid is this, then 17 into 3 I will say. 17 into 3 I will say. What is 17 into 3? 51. So this becomes the answer. Understand the question. The chromosome number in a gamete is given 17. That is haploid number. Okay, gamete is always haploid. It has always haploid number of chromosome. Now, the primary endosperm nucleus, it has triploid number of chromosome. Right. So triploid means triple the amount. So 17 into 3, 51. An interesting question again. Right. So that's there. Next question. During an excavation of soil, pollen fossils were retrieved from deepest layer of soil. The pollen grains remained as fossils because of. Why do you think pollen grains can be still remain as fossils? What is that unique quality about the pollen grains? So pollen grain structure, I will tell you. Pollen grains looks like this. It has a plasma membrane. Outside the plasma membrane, it has something like this, which is known as intine. Okay, which is made up of cellulose plus pectin. Now, Outside this, there is another layer which is known as exine. Okay, what is it known as? It is known as exine. And the exine is actually a tough layer because it is made up of something that is known as sporopollenin. And sporopollenin, please remember, it is the most important thing because it is resistant to enzymes. No enzymes can work on it. No alkali, no acids can work on it. Okay, so this is the reason why the pollen grains remain as fossil. Now, since it is a tough layer and you need to form pollen tube later on, you have certain space left. This is known as germ pore. So in the exine is discontinuous because it has germ pores. Intine is not discontinuous. Okay, from here intine and everything comes out to form the pollen tube and stuff like that. Now the pollen grain, it has two cells initially. Okay, one cell is this cell which has irregularly shaped nucleus. You call this cell as generative cell. Ayo, not generative cell, vegetative cell. What do you call it? Vegetative cell. Now inside the cytoplasm of this vegetative cell floats another type of cell which is spindle shaped, and it will have elongated nucleus also. This spindle shaped cell that you see is known as generative cell. This is known as generative cell. It has elongated nucleus. This generative cell, both of them they are haploid. Only. This generative cell later undergoes division to give rise to two male gametes. So usually in 60% of the flowering plants, angiosperms, the pollen grains are released like this, where it has one vegetative and one generative cell. Later on when it reaches there, it will divide. Okay, and rest 40% are the ones which will already divide itself, and hence it will be in 3-celled condition. If it divides into two, then this is 2 cell and this is 1 cell, right? So 1, 2, 3 cell condition will be there for 40% of the angiosperms. Rest 60% basically they are released in 2-celled condition only. Later on they will be dividing them to male gametes. Please understand that, the question will be coming. So during an excavation of soil, why do you think the pollen grains remained as fossil? Pollen grains are asexual reproductive structures? No. The intine of pollen grains is made up of pectin. That is true, but that has no reason for it to survive during so many years, right? Exine has a spiny ornamentation? No. The exine of pollen grains is highly resistant to enzyme because it is made up of sporopollenin. Works easy. Next, identify the mismatch. PEN is triploid. Yes. Antipodal is haploid. Yes. Zygote is diploid. Yes. Synergids cannot be diploid. No. Synergids cannot be diploid. These are normal cells which are present in the embryo sac. Synergids, egg cell, polar nuclei, and then antipodal cells, they all will be haploid. So this is wrong. It has to be haploid. This came in 2020. Cool. Next, in some plants, stigma and anther mature at different times because. Why do you think in some plants stigma and anther mature at different times? Okay, if stigma and anther is maturing at different times, that means the pollen grains should not be coming over here and fertilizing. To avoid fertilization, this is happening. If fertilization has to happen, then both of them mature at the same time. They do not want fertilization to happen. Okay, so it attracts pollinators? No. It facilitates self-pollination? No. It prevents cross-pollination? No. It facilitates cross-pollination. Now at this point, when they are not maturing together, obviously other pollen grains from other plants can come, and hence it facilitates cross-pollination. It's an outbreeding device. This thing is an outbreeding mechanism. Outbreeding as in to promote outbreeding. As in breeding from other plants. To promote that, this is occurring. This is known as dichogamy. What do you call this as? Works? Next. Nowadays, agricultural practice is expensive to the farmers as they need to purchase hybrid seeds every year. Okay. Agricultural practice is expensive. Why? Because they have to purchase hybrid seeds every year. Which of the following strategies can be employed to overcome this problem? So, instead of fertilization to happen naturally, why not we help in the production of apomictic seeds? If production of apomictic seeds are occurring, then I can overcome this challenge, because in apomixis you do not need fertilization to happen. What happens is that you studied there's a ovule, inside the ovule there is embryo sac, and there are three cells over here. One cell get fertilized, but over here I am saying without fertilization this egg cell forms zygote. This is miracle, but it happens. Yes, it's plant miracle that it happens, and this is known as apomixis, where basically the egg cell is going to form the zygote without fertilization. And when it does this, what happens? This zygote which will be produced will be exactly like the female parent. So if female parent was really good, I had all the features, all the hybrid features already put in there, and I'm getting a zygote just like that. Why will I need to have fertilization and things like that happening? Why should I wait for that? This will naturally occur, apomixis. So production of apomixis seeds is the best way to prevent this, to overcome this. Works. This came in 2019. Now, identify the correct order of steps involved in artificial hybridization in plants. Artificial hybridization as in, I am not allowing naturally fertilization and pollination to happen, or basically pollination to happen. Okay, I am putting my own pollen grains, the pollen grains I want over there. So first thing that I do is emasculation. Okay, suppose this is the flower. Okay, and I have the stigma, style, and the why? I think the food that I ate has been used up. Okay, this is the stigma, this is the style, and this is the ovary. Okay, or probably I should not be drawing like this, I should be drawing properly. I know how much discomfort it might cause you, and I should also be showing that I am not really bad at drawing, but I sometimes do not wish to draw too much. So children, okay, over here, if I do not want the pollen grains to come and land over here, first thing that I should be doing is remove them from here. So I have to remove these guys from here, and this is known as emasculation. Cool. The next thing that I will be doing is I would be bagging this. I will be covering this particular stigma up so that no other pollen grains can come over here. Okay, so that no other pollen grains can come over here, and this particular stigma could attain its receptivity. I will give some time for it to be receptive also, right? Then what will I do? I will remove this. I will take the pollens that I have and I will put it over here. This is known as, first thing bagging I did, then I did artificial pollination. Artificially I pollinated it, and then I rebag it so that no other pollen grains can come again, and basically this pollen grains which are there can fertilize and give rise to the seed that I want. This is known as rebagging. So these are the steps of artificial hybridization in the case of plants. Okay, works. Next question. Testa and tegment of the seed coat represent. So ovule becomes the seed. I told you, ovule, it becomes the seed. So the integuments which were there surrounding the ovule, remember, they become the inner one. The inner one is known as tegment, and the outer one, it is known as testa. So testa and tegment of the seed coat represent dried integuments. The integuments are the one that gets converted to this. Works. Next thing. Among the following statements related to pollens, choose the correct one. In 40% of the angiosperms, pollen grains are shed at 3-celled stage. I told you, yes. Okay, the vegetative cell is there. Generative cell divides into 2, and then you have 3 cells. 1, 2, 3. 2 male gametes and 1 vegetative cell. So in 40%, this is the case. Intine is made up of cellulose and pectin, and it is discontinuous with germ pores. Is it discontinuous with germ pores? No, children. Intine is made up of cellulose and pectin, but it is not discontinuous with germ pores. That is exine, the outer layer, which is discontinuous because it has germ pores. So statement 1 is correct, statement 2 will be wrong. Yeah, statement 1 is correct and statement 2 is what? Incorrect. Cool. Next, when pollen grains is shed at 3-cell stage, name the cell it contains. 3-cell stage as in, I told you, one among them will be vegetative, and the 2 cells which are formed over here are the male gametes. So 1 vegetative cell and 2 male gametes becomes the answer. 2018 question. Okay. Next, even in the absence of pollinators, a assured seed set will be there in what type of flowers? So pollinators are not there, as in wind, water, nothing is there. Still, I will be assured of pollination will be happening. I'm sure pollination will be happening in what type of plants? In the type of plants who will be having their flowers somewhat like this. So the petals basically are going to cover up like this. They would never open, and inside this you will be having the female stigma and everything, and the male stamen also. This type of flower which is always remaining close. So stigma and style both are, sorry, the stamen and the pistil both are here. So pollen from here will be going and landing on the stigma. So I'll just zoom it in and show it to you. So pollen from here will be going and landing on the stigma. So this type of flowers, no, it is known as clistogamous flowers. What do you call it? Clistogamous. Cl stands for closed. Always remember. Okay. And there is chasmogamous flowers which are always open. So in clistogamous flower, you will always see sure seed set. Always seed will be formed. Okay. And they will definitely show just autogamy. There is no other option. They will always show autogamy. There is no geitonogamy, there is no xenogamy, nothing. Always autogamy. Works. Chalo. Next. In a tetraploid plant, if a tetraploid plant contains 48 chromosomes in its nucleus, 48, and this is what tetraploid has been. It has 4 n number of chromosomes. Okay. The number of chromosomes in the egg cell and in synergid respectively will be. So if nucleus, the cells of the nucleus had 2n chromosome, okay, then the egg and the synergid and everyone had n, that is half of it. Agreed? So if now the nucleus cell is 48, I'll just half it. What is the half of 48? 24. So my egg cell will have 24, my synergid will also have 24. I just don't have to see tetraploid, diploid, things like that. It is mentioned that it is 48. Half the number of chromosomes will be there because it has to undergo meiosis. And meiosis, half the number of chromosomes will be there. So it will be 24. There is no other option. So if a tetraploid plant contains 48 chromosomes in its nucleus, the number of chromosomes in the egg cell and in the synergid respectively will be 24 and 24. As simple as that. Works. Okay. Next, pollen grains are generally spherical, measuring about, what is the size? 25 to 50 micrometers. That's a direct question that is there. Okay, what is it? 25 to 50 micrometers. So pollen grains are generally spherical, measuring about 25 to 50 micrometers. 2017 question. Okay. Next, which of the following character is not required for autogamy? So autogamy, as I said, if you do not know, autogamy is basically when there is a flower. Okay, I'm not making the flower again. There is a flower. Where is the flower? From here to here, if things are going in the same flower itself, this is known as autogamy. The same flower, it has to happen. Okay, it is known as autogamy. So question is, which of the following character is not required for autogamy? Okay, so flowers require synchrony in pollen release and stigma maturation. Yes, then only this can happen. Anthers and stigma should lie close to each other. Yes, if they are very far, they won't come together. Flowers should be bisexual. Flowers should be bisexual, as in there should be both the sexes in the flower. Yes. Requires pollinating agents? No, children. It does not require pollinating agents. It is not necessary. Okay, because they are kind of very close. So this is the option that becomes out. Is not required for autogamy. This is the thing. Okay, because they are very close. So why do you need it? 28. In a diathecous anther, each pollen sac contains 1000 mmC, microspore mother cell. What is the total number of pollen grains produced by the anther? So 1 mmC produces 4 pollen grains. So 1000 of them will be producing how many? 1000 mmC will be producing how many? 1000 into 4, which is 4000. Is it 4000? Is it 4000? Tell me. In a dithecaxanther, each pollen sac is mentioned. Each pollen sac is mentioned. And how many pollen sac do you have, children? You have 4 pollen sac. Pollen sac, as in you're talking about microsporangia. You're talking about microsporangia. You have 4 of them. So in 4 of them, each, you are having 1000. So 1000 into 4 is 4000. Agreed. 1000 into 4 is 4000. But what if you have 4 of them like this? So it will be 16,000. Okay, 1 mmC gives you 4 pollen grains. 1000 mmC will be giving 1000 into 4 pollen grains. Okay, that is 4000 pollen grains. But what if you have 4 of them like this? So again, 4000 into 4, you get 16,000. This becomes your answer. Cool. Next question. Which one of the following statement is correct? Okay. Chasmogamous flowers always exhibit gytenogamy. What do you mean by chasmogamous? I told you. Chasmogamous are the ones that are open. So will they always exhibit gytenogamy? So for example, this is a chasmogamous flower. It has the anther and filament, and also the stigma, style and ovary. Over here, this can go from this to here. This is autogamy. So it is not like always they exhibit gytenogamy. What do you mean by gytenogamy? Gytenogamy basically means from this flower to another flower of the same plant. From this flower to another flower of the same plant. So basically, yes, this can happen, but this is not always true. This can also happen. Autogamy can also happen. And from other flower also, this can come. Xenogamy can also happen. This is known as xenogamy, where from other plant the pollen grains are coming. They have to be the same species, that's all. Okay. Now, clistogamous flowers always exhibit autogamy. Is that true? Yes. They are closed. They will always exhibit something that is known as autogamy. This is entirely correct. But let's read the other thing. Chasmogamous flowers never exhibit autogamy. No. Clistogamous flowers exhibit both autogamy and gytenogamy. No, children. It is always closed. So how can you exhibit gytenogamy? It has to be just autogamy over here. So autogamy means within the flower only, if things are happening, it is known as autogamy. Gytenogamy means within the plant different flowers are involved, that means gytenogamy. Xenogamy means 2 different plants are involved. Okay, so in which one will you find genetic variations? You will find genetic variations in xenogamy, because over here you are using a different plant. Over here it's the same plant only. So in gytenogamy and in autogamy, you do not see any genetic differences. It will be all the same. Works? Okay. Next thing. The last question, okay. The 2000-year-old seed excavated from King Herod's palace at Dead Sea belonged to what? The 2000-year-old seed excavated from the King Herod's palace at Dead Sea belonged to 2000. I already told you it is Phoenix dactylifera. Okay, it will be Phoenix dactylifera which will be doing this. If it was 10,000 and it was in the Arctic region, it is Lupine arcticus. Okay, if it was 10,000, that is more than 2,000 actually. So anytime that question can also come, so be prepared for that. So that's all, children. That's all about the questions from sexual reproduction in flowering plants, and I hope that this series is really helping you well. I'm trying my level best to explain things in a better manner to you. Okay, and there is PU2 series and everything going, so if you want, you can watch the video from there also for understanding concepts better. Okay, I hope that helps, and if you have any good things to say or any bad things to say, definitely do not forget to put it down in the comment section. We are all ears. Okay, we are all ears. We are ready to hear you guys, and definitely subscribe to our channel if you think that this particular video or any certain videos are useful for you. So bye bye, take care.